One of the most common questions I get asked on YouTube is funny how I keep starting a lot of my videos like this lately, is whether use of blood with spike protein in it is safe for something like transfusion, whether this is post gene therapy vaccination or post infection. And I haven't had an answer to that until now, because interesting paper came out not long ago recently that actually addressed this very topic, the safety of blood transfusion with products, gene products post vaccination, such as spike protein or the actual genetic material. And I gotta tell you, this paper kind of read like something out of this world in a way. And yeah, that's what we're gonna go through. My name is Dr. Mikola Rashik of Genomics. Let's get started. And first of all, I gotta tell you, this is not a peer-reviewed paper at the moment, so it's preprint. So that always questions the validity of the publication to some degree. So keep that in mind. So it still has to go through that peer review process and basically whether the conjectures made in the publications are legitimate or not. So we're gonna basically address that. But this is not a research paper, it's more like a description paper. The authors talk about what are the risks associated. The goal was to determine what are the potential risks associated with blood transfusions when you have spike protein or vaccine genetic material in it. And I gotta tell you, I was a little bit disappointed because that wasn't, in my opinion, very well addressed, because, or it was, but it's all hypothetical. So it's not like it gives us any extra information in terms of is there any danger or not. However, authors call, I can tell you that the summary is that authors call for looking in the blood as potentially very dangerous, and they're going to make a number of different suggestions in terms of what we should do to protect ourselves. All right, so first of all, they talk about what is being done right now. And the answer is nothing. It's completely assumed that it is safe. Nothing is being screened. Blood is not being screened for whether it has spike protein or the genetic material in it or not. No history is being taken in terms of who took what kind of shots that might result in blood contamination. And it's completely assumed that it's all safe. So we just go on our merry way as if nothing has ever happened. And there is very limited studies showing whether anything has ever been done to address this and test this, whether anyone has actually, let's see if I can catch the skier, if anyone has ever actually looked at this from a scientific point of view. And there is some very limited work indeed. Let's do it this way. Some limited work indeed. And one, for example, group of authors published saying, hey, it's all fine, it all should be fine. And however, all good to use blood post vaccination, COVID-19 vaccination. And the reason why is because, according to the author's summary, no serious events or problems were observed soon after mass vaccination has started. The only issue with that is that obviously a lot of problems have been observed since then. Okay, so, and we're gonna talk about that. So basically that recommendation was based on no data. Okay, and there is apparently also one study that investigated blood post COVID-19 vaccination for its safety for transfusion, and it was determined that it to be safe. And however, this was specifically not whole blood or red blood cells, but plasma or platelets. And again, this was for the safety was measured for a very short period of time. It was only for 30 days, so no long-term data exist. So, and that's it according to these authors' summaries. So we're missing anything more than that. So the authors propose, well, we clearly need more data in terms of safety, and why. And this is where now it gets a little interesting. So Number one, first of all, how is the gene therapy products distributed? First, it was assumed that they would stay localized in the site of injection, basically your arm. That turned out not to be true. We now know that after vaccination, the gene therapy products can be distributed throughout the body because they can get in the blood. So that's number one. While we don't actually have human studies proving this, we have animal studies, but it has been observed that gene therapy products can be observed in other areas of the body. So for example, remember we did a video, for example, on breast milk. And another one has been shown that once your cells take up the vaccine, the lipid nanoparticle, and then the genetic product is distributed inside the cell will start producing spike protein. The spike protein can go on the surface of the cell. These cells can then start releasing these spike proteins into circulations via exosomes. And remember, we did a very large series on the possibility of shedding, and so we discussed exosomes heavily in that particular series as well. So that's known that the spike protein post-vaccination could be distributed throughout the body via exosomes, and that's been already documented via blood studies as well. So that's issue number one. And then issue number two is the fact that in the few years since mass vaccination have started, a lot of information, according to how the author stated, has been documented in published literature of blood-related problems. And maybe, I suppose, let's see if we can catch that. Nope, too late. And maybe the most common one is thrombosis, or basically formation of clots. And I like basically how the authors refer to the post gene therapy vaccination. They called it post-vaccination syndrome. I've never heard this term before. It's the first time I see this. I'm not sure, excuse me, if the authors coined that term or not, but I've never seen it, and I thought this was a very cool term to describe potential issues post-vaccination. And I mentioned that in those last few years, several hundred papers have been published documenting a variety of different problems related to post-vaccination. And let's see, wicked. And their conclusion is that it's undeniable that the spike protein itself is toxic and it's problem. And overall, that means we need to start monitoring for this. But the first issue that they addressed is not necessarily yet the spike protein itself, but in terms of how vaccination could be affecting infections and the risk of infections. So, and I thought that was really interesting. So let's talk about that. So what are we talking about? Well, first they mentioned is that you can produce what is referred to as antibody-dependent enhancement of infection. So you can have antibodies that will literally, instead of helping you, they will actually increase the likelihood of infection. So there's that issue, number one. And number two, as you already know, I am very dedicated to documenting this series of constantly published information that mRNA vaccinated individuals now produce IgG4 antibodies against the spike protein. And the authors mentioned this. So, and I made a series of videos because I basically focus on every paper that comes out on this topic. And the authors mentioned, look, these antibodies, IgG4 antibodies, they basically help to mask the pathogen effects of the pathogen. And therefore, as a consequence, it can actually mask the infection itself from the immune system to a degree, not just for the SARS-CoV-2 virus, but even for other pathogens as well. What that means collectively, those things collectively mean that there is a potential possibility that blood of people who receive the gene product vaccination, this is how they refer to to that in the paper. Such blood runs the increased risk of being infected. And they even mentioned that reports of viremia have been documented as well. And this means that individuals, the medical staff that handles blood from such individuals, are at increased risk themselves. And as a consequence, we ought to be handling the blood with such risk in mind. All right, so then they also mentioned the number one thing that people should be cognizant is that we need to start making our medical staff aware of this risk all over the world, that such a potential risk exists and that we should take appropriate actions when it comes to handling blood products from such individuals. So that's the number one most important message that they mention. Next they mention is that all blood that is being now received, that is to be used for transfusion or basically donation to other people, all blood should be tested for either spike protein or the genetic material of either of the vaccine for COVID-19 vaccines. And the first such test that should be done, that they propose, is direct mass spectrometry that can detect the protein levels. You can even use this type of technology to determine whether there's a vaccinal spike protein present in the blood or natural viral spike protein. We did a video on that topic as well, how you can differentiate between those two. They also propose that we can use antibody assays, where antibodies are connected to specific chemical enzymes that will result in a specific chemical reaction once the antibody binds to the protein of interest, in this case the spike protein, and that's how you can detect it. This is called ELISA assay. So they propose we should be doing that. They also propose we should start looking for exosomes, so we should be screening for exosomes with spike protein in them as well. This is similar to what is already being done in cancer as well, where it comes to screening for exosomes in cancers. So we should be doing that. And of course, PCR to look for the genetic material itself. So all of that should be done to determine, is the blood contaminated or not? Now, what happens once the blood is determined that it is contaminated? And this is the crazy part that completely surprised me. I was like, wait, what? And they said basically such blood should be discarded, thrown out. And if you wonder like, oh my God, why? It's because they said, look, at this point, there's absolutely no known way of how to decontaminate the blood from either the genetic material or the spike protein. And because spike protein, besides the issues that I already mentioned, another problem with the spike protein is that it can form amyloid-like maybe structures, I should say. We did a big series on that as well, when it comes to formation of microclots with combination of the spike protein. And when it comes to amyloid-like protein, which means proteins that are deformed in certain ways, they can be very resistant to degradation. We've actually discussed that in that series as well. And that really could make it impossible at the moment to clean up the blood. There is literally no way of knowing at the moment how can we clean up the blood. And because it poses a risk, such blood should be discarded. Crazy. Like I said, it reads like a paper from another world, because I've never heard anyone discuss this. And by the way, I believe these authors were from Japan. I hope my memory serves me well. Now, if hospitals cannot afford to discard blood themselves and they have to use it because, of course, of limited supply, then what the authors propose is that such information that they're using contaminated blood needs to go into transfusion informed consent for the donor, and the donor should be informed of the potential risks and that such a contamination could pose to such individual. And this should now be adopted as a standard. Now, of course, they mention is that we need to develop international bodies that would start measuring what are the risks, determine what are the potential risks. So this needs to be studied in detail. And on top of that, once these risks are determine what are the safe levels of these products, either either the genetic material or the spike protein, what are the safe levels in the blood, genuinely safe levels in the blood for blood to be used. And then of course also these bodies should determine how such blood should not be contaminated in the first place as well, like how to limit contamination of blood with these unwanted products. So all of that should be determined. And they even mention is that, this is really interesting, is that this should be entrenched in legal law. So the pathogenicity component of the spike protein should be entrenched, legalized. Basically, should be legally determined the actual pathogenic status of the spike protein, and laws should start incorporating the information how to protect individuals from not being contaminated with the blood from those individuals who had either spike protein or genetic products down the road. And basically this we need to determine. So they're really going far with these suggestions. Now, another thing that they mention is the possibility of autoimmunity. They mentioned that frame shifting has been documented recently using the mRNA gene therapy. And basically frame shifting means is that the genetic code is not used properly. It's the machinery, molecular machinery that is supposed to be decoding that genetic code can latch on in the wrong place and basically produce something completely different than spike protein. We need to determine whether this really happens or not. If it does happen, such abnormal products could lead to autoimmune conditions. And they mention also overpresentation of the spike protein to the immune system on the surface of the cells, which is one of the reasons why they claim we're seeing these IgG4s. That in itself could be leading to autoimmune conditions. So also as a consequence, they mentioned that those people whose blood is determined, once they donate the blood and it's determined that they have spike protein in the blood or genetic content, such individuals should also be informed about the potential risk of developing autoimmune disease as well. And then finally, the authors mention, hey, what do we need to do now? The final call to action is to stop using the gene therapy products themselves. So stop vaccination. The reason why is because we need to stop contaminating the blood until safety is established. And they're saying, look, we also need to address that there are real problems right now when it comes to blood issues. We got to stop mucking around, pretending as if nothing is happening. And we need to seriously address this issue and seriously start doing something about it. And they're saying, like, we need to start doing this now. So that's the summary of this paper. So really, I was a little disappointed in that. Doesn't seem like there's any viable solution right now. As I mentioned, how can you get rid of blood when you need blood donations? There's always seems to be shortage of blood donations, and the bulk of the world is now vaccinated anyway. So I wish they actually spend more time into looking at the possible solutions for how to maybe fix the issues if they think these are such serious issues. Because as I mentioned, a lot of this is theoretical in terms of whether this is a serious problem via transferring of these products via blood from one person to another. And until it's proven, this is potentially sounds very scary, but time will tell. Anyway, it's the first paper I've seen that finally called to action in terms of looking at the safety of blood in general and why we should be looking specifically for uncontaminated blood and determining that. I do agree though with the concept that we should be screening blood for presence of spike protein, because clearly spike protein is not nothing burger. It's definitely can create some issues. As I mentioned, we did a video on even the potential problems with clotting, and that's not irrelevant. So I agree with that. All right, I'm gonna pause it right here. Hopefully the wind will allow this video to survive. And listen, please subscribe to the channel if you haven't already. Please leave us a comment. Please give us a like. This is how we grow, so we can continue studying and giving you more content like this. And also, please check out my Patreon account where I post additional content that does not make it to the YouTube. And I look forward to seeing you in the next video. And as always, make sure you get outside. Bye, everyone.